Today's practical is on photosynthesis, which is one of the required practicals for biology. Photosynthesis is a vital process by which green plants use carbon dioxide and water in the presence of light to make sugar. Now, green plants use sugar as a food source to provide them with energy for growth and to produce oxygen. In this practical, we will be looking at the effect of light intensity on the rate of photosynthesis by measuring the oxygen production in pondweed and counting the number of oxygen bubbles when the pondweed is placed at different distances from a light source. So, photosynthesis is affected by factors such as carbon dioxide concentration, water supply and light. Our hypothesis is that as we increase the distance between the pondweed and the light source, the rate of photosynthesis will slow down. And for this practical, we need a boiling tube clamped to a clamp stand, a lamp, a meter ruler, a thermometer, a stopwatch, some sodium hydrogen carbonate solution, and pondweed, which I have here in a beaker of water so that it stays fresh. So, our independent variable in this practical is the distance. Our dependent variable is the number of oxygen bubbles. And the control variables are the temperature, the brightness of the light bulb, the type and size of the pondweed, the concentration of the sodium hydrogen carbonate, and the time scale for measurement. So, firstly, we need to take a piece of pondweed which is about 10 centimeters long. Now, I have cut this pondweed from the top at an angle so that we can get a consistent stream of bubbles. We need to now place the pondweed in the boiling tube and pour in the sodium hydrogen carbonate solution, completely covering the pondweed. Now we need the pondweed to adjust to the environment within the boiling tube. Next, we need to place the lamp at 10 centimeters distance from the pondweed. So I will use a meter ruler, and where 10 centimeters is, I shall position the lamp. Now, there are errors associated with measuring the distance between the lamp and the pondweed. So, to overcome this, what we should do is always measure from the same position each time we change the distance. And to be a bit more accurate, what we can do is make sure that the boiling tube is in line with the clamp stand base and that the head of the lamp is in line with its base. So when we measure the distance between the base of the clamp stand and the lamp, we can somewhat be sure that the distance is the same as it is between the pondweed and the lamp. Now I can turn on the lamp. We need to allow the pondweed a short while to adjust to the light intensity before we start counting the bubbles. Also, we do not want the temperature around the pondweed to change, so we will put in a thermometer in the boiling tube so that we can monitor the temperature throughout the practical. Now, we are using a normal light bulb, and the problem with this is that these give off excess heat, and they can change the temperature around the pondweed, which would then give us inaccurate results. To overcome this, what we can do is use a glass heat screen in between the both, or we can use a beaker with hot water and place it between the pondweed and the lamp, which will then absorb the excess heat. So we need to now wait till we've got a consistent stream of bubbles to start counting. Okay, we can start to see some bubbles being produced. So I will start the stopwatch and start counting to see how many bubbles we get in 1 minute. It's best to count at eye level for accuracy, and we will just count the bubbles that will emerge from the cut end of the stem. So I will start the stopwatch now. Okay, so for that minute we had 32 bubbles. Now we need to repeat this step 2 more times so that we have readings to calculate a mean value. And we need the mean value for reliability and accuracy of results. After that, we can change the distance so that the pondweed is at 20 centimeters, 30, 40 and 50 centimeters away from the light source. Now, sometimes we can't count the bubbles accurately because they are too fast to count. So what we can do is either video record the bubbles or have more than one person counting. Remember to allow the pondweed 5 minutes to adjust to the new light intensity each time. The results can be recorded in a results table. Our results show that as we increase the distance of the pondweed from the light source, the number of bubbles produced per minute decreased. As at 10 centimeters we had 32 bubbles, and at 50 centimeters we had 2 bubbles, meaning that as we reduce the light intensity, the oxygen production is reduced. The results support our hypothesis. We can then plot a graph with the distance from the pondweed to the lamp on the x-axis and the mean number of bubbles per minute on the y-axis. The bubbles produced are not always the same size, so it's not a good indication of the amount of oxygen produced. So, to achieve more accurate results, we can carry out another practical in which we can collect the oxygen produced and measure its volume rather than counting the bubbles. To do this, we can use a setup in which we have a large beaker of water, and in this we place a piece of pondweed under a glass funnel and place it in the bottom of the beaker with the spout pointing upwards. We then place a measuring cylinder upside down over the spout of the funnel. After placing it near a light source at different distances, the bubbles of oxygen will get collected in the measuring cylinder, and you can read off the volume for each distance. We can also control the temperature in this way, as the large beaker acts as a heat shield.